an exam paper with blanks. The blanks sat on the blank. To fill in cat and mat, you need to understand grammar, vocabulary, and the patterns of natural language. No teacher told you the answer. The structure of the sentence itself constrained the possibilities. Self-supervised learning works the same way. The data creates its own training signal. No human labels needed. Prediction task forces the model to learn useful representations. Supervised learning requires paired examples. Is protein sequence, has this function, this tissue image is cancerous, this molecule binds with this affinity. Each label costs expert time and experimental resources. A pathologist might annotate 1,000 histology images in a month. A chemist might measure 100 binding affinities in a week. But unlabeled data is almost free. 250 million protein sequences in UNIRF, 30 million cells in Celogen, millions of histology images in digital pathology archives. Self-supervised learning exploits this asymmetry. Core idea: create a prediction task where the label comes from the data itself. Take an input, corrupt it in a known way, mask some tokens, add noise, remove patches. Ask the model to predict the original from the corrupted version. The original data is label, free, abundant, and automatically generated. The model can't memorize the answers because each training example is corrupted differently. It must learn general patterns, the structure of protein sequences, the relationships between genes, the morphology of tissues to solve the prediction task. Masked language modeling is the original self-supervised blockbuster. BERT: take a sentence, randomly mask 15% of the tokens, predict the masked tokens from context. ISM applies the same idea to protein sequences. Mask 15% of amino acid positions and predict them from the remaining sequence. To predict a masked leucine at position 47, the model must learn which amino acids co-all with this position across evolution, which residues make March 22nd, 2026 context with position 47, what functional motifs include this position. Prediction task forces the model to discover protein biology. Next token prediction is the autoregressive alternative. Instead of masking random positions and predicting them all simultaneously, predict each token given only the preceding tokens. EVATE: given the cat sat on, predict the scGPT. Given the expression levels of genes 1 through 50, predict gene 51's expression. The model sees context only from the left, it can't peek at the future. This generates naturally, sample one token at a time to produce new sequences. The trade-off: MUM sees bidirectional context and is better for understanding. Autoregressive sees only past context, but can generate. Masked autoencoders extend masking to images. Take a tissue image, divide it into patches, mask 75% of patches, far more aggressive than BERT's 15%. The encoder processes only the visible patches. The decoder reconstructs the full image from the partial information. To reconstruct the masked regions, the model must understand tissue structure, cell morphology, spatial organization, and staining patterns. He et al. Showed in 2022 that this aggressive masking is not only sufficient but optimal. It forces the encoder to learn meaningful representations rather than interpolating from nearby pixels. Masked language modeling: hide tokens, predict from context. Next token prediction: predict the future from the past. Masked autoencoders: hide patches, reconstruct the image. Each creates labels from the data's own structure. No human annotation. No experimental measurement. The model teaches itself by solving prediction puzzles. Next, another self-supervised paradigm: learning from similarity.